Okay, good evening all. Okay, okay, good evening everyone. Good evening, good evening. Good evening all. Okay, what about others? Or what happens? Very less number of students today. Okay, good evening all. Good evening, good evening. Good evening all. Okay, happy Women's Day to all the women's. Good evening all. Okay. So friends, let's continue. So friends, in the day before yesterday class, we were discussing about the DNA replication. If you all quickly recall friends, if you all quickly recall, we understand the different type of rules of the DNA replication. And if you all remember, we were about to read what friends? We were about to read this particular semi-conservative model. If you quickly recall friends, we understand there are different rules of the DNA replication is there. Semi-conservative, start at the origin. The process of synthesis of a new strand always takes place at 5 prime to 3 prime direction. It can be unidirectional or bidirectional, then followed by semi-discontinuous. RNA primers are required, high fidelity. So friends, I am requesting all of you people, wait for 15 minutes. Okay, let me explain the entire experiment little elaboratively. After that, I will solve all your doubt. So requesting all of you pay attention towards the lesson. Let's focus friends, easy to understand. Okay, first, what is a semi-conservative model? I hope friends all of you remember very clearly. Semi-conservative model, what I said to all of you, according to the semi-conservative model, two parent strands are get separated and each parent strand acting as a template strand, and friends there is a synthesis of the daughter strand. That is what exactly we can call semi conservative model. If all of you quickly recall, in the last class I told you friends very clearly who proposed this particular semi-conservative model. Watson and Crick proposed. I told you na 95% of scientists accepted this model, but few of the scientists opposed this model. Because why they have opposed this model? Because there is a no structural proof, there is a no experimental proof to prove the semi-conservative model is the idealized model for the DNA replication. All of you listen to me friends. So by good evening all, please continue, please listen to me for 15 minutes continuously. Friends, I will explain the unit 3 very elaboratively and very depth. I only expect your great patience and interest. So by opposing the semi-conservative model, conservative model is proposed, as well as friends dispersive model is also proposed. What I said about the conservative model, all of you pay attention, just listen to me. According to the conservative model, the entire parental DNA, listen to me friends, according to the conservative model, entire parental DNA is preserved and converting into daughter strand. What about the dispersive model? According to the dispersive model, it's like a cut and patch, cut and patch, cut and patch, cut and patch, which means small daughter parent, daughter parent, daughter parent, daughter parent, daughter parent. That is what model friends? We generally call it as a dispersive model. Right. Now friends, which model we have to prove now? Semi-conservative model we have to prove. Everyone pay attention and listen to me slowly and carefully. Pay attention. See friends, what is the ultimate goal of the semi-conservative model? According to the semi-conservative model, we have to prove there will be a two parent strands are always associating with the daughter strand. In a simple line, my friends, in a simple words, we have to separate the parent strand and daughter strand. Always every student remember, parent strand is the heavy strand, daughter strand is all the time light strand. This is what we have to prove actually. Now listen to me, how we have proved. Two intelligent scientists named as Matthew Meselson and Franklin Stahl, they proved semi-conservative model is the idealized model for the DNA replication. Now all of you pay attention, just listen to me carefully. First, what they have done? First, they have taken a test tube. Listen friends, every one of you. First, they have taken a test tube. In this test tube, E. coli was kept. And where this particular test tube was kept? In the heavy nitrogen. From this, they have isolated a particular DNA. And my friends, please listen my words. When they isolated the DNA, they start observing what, my friends? They start observing the base. When they start observing the base, what they find? They find heavy DNA. Now next, what they have done, once again, good evening everyone, please continue. Once again, they have taken the test tube. This time, my friends, you know what they have done? In the test tube, they kept E. coli culture, but where? In the N14. And once again, from the test tube, friends, they have isolated the DNA. After isolating the DNA, what they have done? Same way, they have observed the base. My friends, when they observed the base, what they found? They found light. But what is our semi-conservative model? I said it to all of you. According to the semi-conservative model, we should contain both heavy strand and light strand. We called intermediate. We called hybrid actually. Friends, all of you pay attention and listen to me very carefully. After some time, what they have done, friends? Once again, they have taken another test tube. Friends, you know this time what they have done in this test tube? They have added equal concentration of N15 they have added, and friends, they have added equal concentration of N14. So along with the N15 and N14, my friends, what else they have added? They have added the cesium chloride. Now you can ask me a question, sir, there are many salts are there, why they have used only cesium chloride? What is the reason behind that? The reason, friends, the density of the DNA and the density of the cesium chloride, which is extremely equal, which is 1.91 gram per centimeter cube. This is the same density of the DNA and cesium chloride. That's why they have used the cesium chloride. Now in this, what is there? Cesium chloride in this test tube, cesium chloride is there, N14 is there, N15 is there. My friends, what they have done? They rotated at a very high speed for the period of how much time? For the period of 20 hours. Once they rotated for the period of 20 hours, what is the point you people has to understand? Based on the density gradient, what my friends? Based on the density gradient or based on the weight variation, all got settled. Now you only tell me, friends, among these which is having highest weight? Cesium chloride having the highest weight. As a result, friends, cesium chloride will settle down. After the cesium chloride, which is having the weight N15 will settle, then followed by the N14, because N14 having a very light weight. My friends, now based on this little understanding, Matthew, Meselson and Stahl, they come to a conclusion. What is the conclusion they come? They have come to a conclusion that we can easily separate. What we can easily separate? We can easily separate the parent strand and the daughter strand exclusively based on their density gradient. What, friends? Based on the density gradient, we can easily separate the parental strand and the daughter strand. They understood that. Based on that simple logic, now they started studying what, friends? They started studying the semi-conservative model. You know what they have done? Let me tell you first. First, they have taken a bacterial culture. which is kept where? In the N15. Okay, and when they have isolated, what they found, my friends? They found completely heavy DNA. You can see it. Completely heavy DNA. Now, my friends, you know what they have done? They have transferred this particular N15 to what, friends? They have transferred to the N14. After transferring the N14, what they have done, for every 20 minutes they are taking the sample. Good evening everyone, please continue. Every 20 minutes they are taking the sample. Whenever they are taking the sample every 20 minutes, first 20 minutes you only try to understand what they got. They got 100% heavy. They got 100% heavy. Next generation we got 50% light and 50% intermediate. My friends, what do you mean by the word intermediate? Intermediate means, always remember, my friends. I'm so sorry, friends. In the first generation, they got intermediate, which means they never get either heavy nor light. What they got? Intermediate. Intermediate we can also call as a hybrid. What is hybrid or intermediate? Combination of the parent strand, combination of the daughter strand also. Now, second generation we got 50% intermediate, 50% light. 25%, 12%, 6%, so on. It is getting repeated. So friends, we can easily come to a conclusion, no? So second generation is sufficient actually to prove semi-conservative model only the idealized model for the DNA replication. Now, what is intermediate or hybrid? What I said to you, it is the combination of daughter strand, parent strand, daughter strand, parent strand, daughter strand, parent strand. Every time it is getting repeated. Suppose we have to disapprove. Suppose example, you only try to understand if it is a conservative model. What did I say to all of you about the conservative model? Entire parental DNA preserved and it is going to be converted into daughter strand. Now you only tell me, do you observe anything like that? We never observed entire parent strand is giving rise to the daughter strand. It is not possible. Now if you look into the dispersive model, what did I said? Daughter parent, daughter parent, daughter parent. It's like a cut and patch. Have you observed anywhere cut and patch? Not at all. So friends, the second generation is sufficient to prove semi-conservative model is the idealized model for the DNA replication. Such a simple model it is. So friends, some students will get confusion. I am repeating for every one of you. Just quickly understand, very quickly, what we have done. What is the ultimate goal of the semi-conservative model? According to the semi-conservative model, we have to separate the two parent strand, and two parent strand should act like a template. There should be a synthesis of the daughter strand. So for that, what the Matthew Meselson did first, they have taken a test tube first with the N15, and from that particular bacterial culture, they have isolated the DNA. When they isolated the DNA, they found the heavy. Then later what they observed? N14. When they observed the N14, they found the DNA which is only light. But what is our goal? We should not have the heavy and we should not have the light. We should have the combination of both heavy and light, which we called intermediate, which we called hybrid. Now what they have done? They have taken a test tube. In the test tube they have taken N15, N14 and cesium chloride equally, and they started rotating. They started doing the process of centrifugation up to 20 hours. How much time? 20 hours at a very high speed. Then what they observed, friends, based on the weight variation, based on the density gradient, all got settled. Which is having the highest weight? Cesium chloride. So settled down. Next N15, next N14. My friends, what's the conclusion? Now conclusion is that we can easily separate the parent strand, daughter strand based on the density, based on the weight. So by this they started proving the semi-conservative model. When they observed the semi-conservative model, in the first generation what they got? They did not get heavy, they did not get light. What they got? Intermediate. What is intermediate? Parent strand, daughter strand. That is only our semi-conservative model. In the generation 2 also we got the intermediate, generation 3 also we got intermediate, generation 4 also we got intermediate. It's keep on repeating. Anywhere you can't get the conservative model, anywhere you can't get the dispersive model. Hence generation 2 is extremely sufficient to prove semi-conservative model is the idealized model for the DNA replication. This is the Matthew, Meselson and Stahl experiment. Friends, everyone say yes or no. Is that clear, everyone? I will put one animation also in the Telegram link. You can watch it. You will feel very easy. Okay. Everyone, friends, quickly give me the reply. Is it clear, everyone? Quickly. You can ask any kind of a doubt. Suraj, I don't know why people are getting link for troubling the PPT. I will show you in this particular class to all of you how to download. It is not at all difficult, friends. I don't know why every one of you saying in the Telegram we are getting difficult in downloading the PPT. Now I am showing to all of you. Listen, look into this. See, I have opened the Google. I don't know how it is difficult for you. See, look into this. Type sciencefestival.in. Now, if you come to this particular home page, you are able to see everything very clearly now. So it is showing an option called as a free downloads. It is showing an option called free download. If you look into the free download, there is a two options are there. One we called PPT 2022, another one is called recorded lecture June. If you click on the PPT 2022, see here, you got the PPTs. Then if you just click on here, you will get it downloaded automatically. I don't know why it is difficult, friends. Now same way, if you click on the recorded lectures of June, now you can see the recorded lecture. See here, everything was very clearly kept. Easy to know it. Why to face difficulty? It's very easy, friends. Okay. If you are getting difficulty, my friends, according to my knowledge, your internet will be slow. That's all. Anyway, let me know if you have any kind of a problem. Because no. Tarun, when it comes to your question about the cesium chloride, the answer for that is cesium chloride we have used, it can act as a salt. We have used only for the purpose of density, only for the purpose of weight variation only we have used the cesium chloride. And Kaushalendra Singh and when it comes to your question, why we have done the comparison between the N15 and N14? Because N15 here it is only representing as the parent DNA, which is the heavy DNA. N14 is basically representing as the light DNA. See, N15 we can call it heavy nitrogen. Heavy nitrogen. N14 what do you call, friends? Light nitrogen. Sadguri, without nitrogen, how can you observe the base, dear? What is our DNA is made up of? DNA is made up of nucleotide. Nucleotide contains sugar, and sugar then followed by the base and followed by the phosphate. Now what does this particular base is contains? Base is nitrogen only, right? We can call nitrogen base, nitrogenous base. So that's what we will use the nitrogen. You understand? Whomever got the doubt clear, let me know quickly. Okay, perfect. Correct, Vishal, it is radioactive and heavy. Good. So friends, I will automatically solve your doubts. You just listen to me. Most important thing I want to say, have trust on me. Okay, I never miss a small thing which is important for the exam. Okay, have that faith on me and try to understand the depth of the concepts whenever I'm teaching. Automatically your concepts will be very, very clear. Correct, Tarun, whatever you said is absolutely right. Correct. Okay. Friends, let's continue. So next one, first rule we have finished, the semi-conservative rule. The second rule, friends, starts at the origin. Okay, listen to me very carefully. So please don't interrupt me for the period of 10 to 20 minutes. I will explain all the rules. I'll give the time. You can ask multiple doubts. I'll clarify every rule, every doubt. So second rule, it will start at the particular point. Now all of you listen my statement. in case of a DNA replication, the process of replication never ever start randomly. Whether it is a prokaryotic organism doesn't matter, whether it is a eukaryotic organism doesn't matter, friends. The process of DNA replication always start only at a particular point. The point at which the process of DNA replication will start, we will be calling it as the ORC. And what are the unique statements? All of you people has to remember, friends. In case of a prokaryotic organism, friends, there is only presence of one ORC. But friends, please listen, because prokaryotic organisms like bacteria are very simple organism. They are not at all the complex organism. So how many total number of base pairs they contain? It contains just one ORC, and it consists of 245 base pairs. And friends, when it comes to the eukaryotic organism, we are very complex organism. So, and you know very well, our DNA is a very long molecule. Our entire DNA is not going to be our entire DNA has to be replicated in the S phase. Now listen, DNA is a long molecule, time is very less. So friends, if we have only one ORC in the eukaryotes, the process never occur faster. So all students keep it in your mind, my friends. In case of a eukaryotic organism, there will be, first of all, we never called ORC in case of a eukaryotes. Instead of calling the ORC, in case of a eukaryotes, we will be calling it as the replicator. Now listen to me carefully. How many replicators are present? Here replicator or ORC is the same, but in eukaryotes we will use the word replicator. Here I have clearly said you that in case of prokaryotes there is only one ORC. My friends, in case of a eukaryotes, all of you listen, 150 replicators will present. Not one or two, 150 replicators are present, because our DNA is a very long one, very long DNA molecule. All of you listen, friends. However, we take all the time for the best example for the prokaryotic organism, we will take E. coli as the best example. Whereas whenever we are reading the eukaryotes, we will always take yeast as the best example. Because you can ask me a question, sir, there are many organisms are there, then why you are considering only yeast? You can take any other, right? You know, right? We are having 23 pairs of chromosome, yeast having 16 pairs of chromosome. So some are less, not equal, some are less equal to the humans. That's why we will always consider the yeast as the best example. So I'm quickly repeating for all of you, friends. You no need to worry, I will explain everything twice, elaboratively, just follow me patiently. So prokaryotes, it contains one ORC contains total 245 base pairs. Whereas in eukaryotes, there are, we never use the word ORC, we will always use the word called as the replicator. So friends, how many replicators are present? 150 replicators are present. I told you na, in case of eukaryotes, we have taken the best example as yeast. Now, what is the statement all of you has to remember for the exam is that in case of a yeast, the replicator has given the another name. What is the name of the replicator in case of a eukaryotic organism? We will be calling it as the ARS, which means autonomously replicating sequence. Autonomously replicating sequence, ARS or the replicator is same. There is no change about that. This is the second rule. And the third rule, 99% of the students don't know about this. Okay, I mean to say I'm talking about this particular rule. How does it work? It is very important, my friends. You have to know it. And listen my statements very carefully. Last and final time I am saying, whenever you are listening my lessons, especially, I will make sure that you will understand 100% of concept very effectively in terms of application. Okay, the only thing I'll expect from you is great interest and patience. Connect to me when I'm teaching, by the concept will be very easy. I always try to involve in that particular concept. Now listen, the direction of the new strand. You all commonly know, right friends? The new strand has to be synthesized in which direction? 5 prime to 3 prime. Now we are going to prove in this particular slide. What we are going to prove in this particular slide? Why the DNA new strand should not be synthesized in the 3 prime to 5 prime. Why it has to be synthesized only in the 5 prime to 3 prime. Now we are going to prove it. Now every one of you pay attention and try to understand every single point. Look into this. First point. What is the enzyme that is involved in the DNA replication? DNA polymerase enzyme. My friends, please listen my statement. The versatile DNA polymerase enzyme, it always read the parent strand. I am talking about parent strand. DNA polymerase enzyme always read the parent strand in 3 prime to 5 prime direction. If it is reading the parent strand in the 3 prime to 5 prime direction, undoubtedly my friends, it is going to synthesize the daughter strand in the 5 prime to 3 prime direction. This is one reason. And another important reason I want you to know, in the last class I told you, how does the phosphodiester bond will be formed? If the phosphodiester bond will be formed, there should be a presence of the OH group. So you know, right, where the oxygen can act as a nucleophile, it will cut the phosphate group, then only phosphodiester bond is formed. So I can tell you in a simple way, friends, wherever there is a presence of the 3 prime hydroxyl group, there only nucleotide is going to be added. Anyway, let me prove. Focus everyone, pay attention. Now this is the nucleotide. Nucleotide contain how many phosphate groups? We already read it very clearly. In case of a DNA, every nucleotide comes with the 3 phosphate. We can call it as a triphosphate. So how many phosphates are there? 3 phosphates are there. Friends, pay attention. Here what group is there, friends? OH group is there. Now what is happening? This oxygen nucleophilically come and cut what? Come and cut here. My friends, whenever it cut, what's happening now? The 2 phosphates are getting released. This is what exactly, friends, we will be calling, by that PPA will be released. Now you only try to understand. Now this OH and phosphate group joined with each other. Now successfully nucleotide is added. There is no doubt about it. Now you all know very well, we're gonna read it also. New strand can be synthesized in the 5 prime to 3 prime. We read very clearly the proofreading occurs in which direction? Proofreading is going to be occurred in the 3 prime to 5 prime direction. Every one of you pay attention. During the proofreading, what is happening? The DNA polymerase enzyme identified. What the DNA polymerase identified? Whatever the nucleotide that we have added, that is the wrong nucleotide. That is not a healthy nucleotide. Now what we are going to do? We have removed that particular wrong nucleotide. Now, even though we remove the wrong nucleotide, no problem. What is the point every student has to remember? Still, there is a presence of OH group. Still there is a presence of the OH group. Because of that particular reason, once again what is happening? New nucleotide will come, healthy nucleotide. Once again OH group is here and it will come and cut this particular phosphate group. PPA will be released, new nucleotide is added. So friends, now what is the final statement I want you to know? The major reason why the DNA strand is synthesizing in the 5 prime to 3 prime direction, because high energy bond is cleaved. You can ask me, sir, what is high energy bond here? Of course, my friends, phosphate group. Because of the presence of phosphate group only, what is happening, friends? The energy will be released, then nucleotide can be added. Once again, I'll summarize very quickly. Focus. First, what is happening? Here the 3 prime hydroxyl group is present, where oxygen acting as a nucleophile, it will come and cut one phosphate group, PPA will be released and the nucleotide is added. But during the proofreading, it was identified that whatever the nucleotide was added, it is the wrong nucleotide. That's why it got removed. But still, what group is present? OH group is present. So another nucleotide once again can be easily added by cleaving the phosphate group. We have proved why the DNA new strand has to be synthesized in the 5 prime to 3 prime only. Now what we have to prove? We have to prove why the new strand should not be synthesized in the 3 prime to 5 prime. This I am going to prove it. Now everyone pay attention, try to understand the depth of the concept now. Now we are imagining everything reverse. You can ask me, sir, does really this thing happen inside the cell? I never say that, because this is the hypothetical. We are proving why the strand DNA daughter strand is not happening or not synthesizing in the 3' to 5'. We are showing it hypothetically. Focus. Now you all commonly know right now, friends, now the synthesis is happening like this. At the 5th carbon, how many phosphate groups are present? We all know that 3 phosphate groups are present. Now you can see, right, this is the new nucleotide. Friends, new nucleotide also contain how many phosphate groups, friends? New nucleotide also contain the 3 phosphate group. Now you can ask me a question, sir, how does this particular nucleotide will added here? I can tell you, this is hypothetical. Anyway, this nucleotide contain OH group, right, my friends? This oxygen nucleophilically will come and cut what? 1 phosphate. Correct now, friends? Then how many phosphates are left here? 2 are left. 2 got released out. Now see here, finally the nucleotide is added. Simple point. Now you can understand easily. Now during the same thing proofreading occur, friends. During the proofreading we identified that what we identified that this nucleotide is a wrong nucleotide. Now what I have done, I have removed the wrong nucleotide. No problem. Now you only try to understand. Now after removing the wrong nucleotide, I have to add the new nucleotide. Now you only tell me, friends, do you think I can add this new nucleotide? No, right? You only try to understand. Here OH group is present, no doubt. But you tell me, if the OH group, if this nucleotide want to add here, there should be compulsory 3 phosphates should be present. Then it will cut 1 phosphate, 2 phosphates will be released. Then only nucleotide can be added. But you only understand, is there any presence of 3 phosphate groups here? Not at all. So what is the statement I can give you? Reaction does not proceed. The reason, high energy bond is not getting cleaved. Once again I'll summarize. Look into this. See here, we are understanding hypothetically synthesis is happening in this direction. So this is the nucleotide which contain OH group. Initially it will go and cut 1 phosphate group, 2 phosphates will be released. Now the entire nucleotide added here. During the proofreading we understand that this nucleotide is a wrong nucleotide. We removed it immediately. But now we are going to add the new nucleotide. But is that possible? Can we add the new nucleotide? Never ever. Because here there is no 3 phosphates are there. That's why I can conclude to all of you, the new strand always has to be synthesized in the 5' to 3' direction only. Never forget that point at all. So that is the rule number 3. Synthesis of a new strand in the 5' to 3' direction. This is an important statement you have to remember. Next rule. Friends, I will explain every concept very depth. Questions won't go beyond that level. Pay attention. Then the next rule is the process of DNA replication will be either unidirectional, friends, or bidirectional. Now the statement all of you has to remember, my friends. Sir, in which organism the DNA replication will be unidirectional? In which organism the DNA replication is the bidirectional? But I want you to know one important point. In case 90% I can tell you, friends, in case of a prokaryotic organism also, friends, even in case of the eukaryotic organism is also, friends, both prokaryotes and eukaryotes, the type of replication is the bidirectional replication only. First of all, look into the picture carefully, understand what is unidirectional, what is bidirectional. Look into this. Unidirectional means, suppose example, friends, this is the origin. From this origin, the strand is moving only one direction. I mean to say the fork. The replication fork is moving only one direction. That is what do you call unidirectional. But if you look into the bidirectional, what you have to understand, see, from this is the origin, there are 2 forks are there. One fork is moving in the clockwise, another fork is moving in the anti-clockwise. 2 forks starts moving in a 2 different directions. That is what do you call, friends, bidirectional. One we called unidirectional, one we called, friends, bidirectional. But what's the common point I said it to you? In prokaryotes and eukaryotes, the most common type of replication direction is the bidirectional only. Now friends, what is the next rule? Friends, still we are reading the rules only. After the rules, we have to read enzymes. After the enzymes, we have to read the process. Then whatever the type of question they may ask, we can understand every question very easily. So pay attention. Next rule, semi-discontinuous replication. Once again, I am reminding to all of you, this is a rule. It is not a process. Okay, what is the semi-discontinuous rule? If you quickly recall, all of you pay attention. This is one parent strand. In which direction? 3' to 5' direction. This is another parent strand. In which direction? 5' to 3' direction. And if you look into our versatile DNA polymerase enzyme, what I said to all of you, it is going to read the parent strand in 3' to 5' direction. Hence, there is no problem here. Now, it is going to synthesize the daughter strand in 5' to 3'. No problem at all. Towards the replication fork. That's why I called this strand as a leading strand. Even I can call this particular synthesis as the continuous synthesis. This is just a rule. I am explaining overview. That's all. In the replication process, we will read very elaboratively. Now, here the another strand is synthesizing like this in the form of a pieces. One is primer Okazaki fragment. Friends, please kindly don't confuse. Sir, what are Okazaki fragments? Okazaki fragments nothing but they are nucleotide sequences only. Based on the name of the discoverer, we called it as what? Okazaki fragments. You can ask me, sir, can you explain little elaboratively why the strand is synthesizing oppositely? That we will discuss when we are reading the process very clearly. It is just a rule, so I am explaining you that the DNA replication has to follow the semi-discontinuous. And one more interesting point is what? Whether it is a leading strand doesn't matter, whether it is a lagging strand doesn't matter. What is the interesting point? Both the daughter strands has to be synthesized in the 5' to 3' direction only. That is the universal statement. What is that? Whether it is a leading strand or the lagging strand, both has to be synthesized in the 5' to 3' direction only. So friends, all of you listen to me. What is the next rule? I can say one of the most important rule. Synthesis of RNA primers. Friends, I will tell you one very good example to you. First listen. What is the function of a DNA polymerase enzyme? We read very clearly that the function of DNA polymerase is what? Adding one nucleotide with the another nucleotide. That is the function. How can I define the word synthesis of a daughter strand? If I ask you, please define me the synthesis of a daughter strand. How can you define? Synthesis means what? Adding one nucleotide with the another nucleotide. That's all. That is what we called synthesis. But friends, if the DNA polymerase enzyme want to synthesize, want to add one nucleotide with another nucleotide, first there should be a presence of nucleotides. When there is a presence of nucleotide, to that nucleotides, the enzyme keep on adding other nucleotides. I will give you one very good example, real example. In the childhood and even nowadays also you might observe in the villages, we are having borewell pumps. What pumps? Borewell pumps. What is the speciality of a borewell pump? If we want to get a water from the borewell pump in the villages, what we use it to do? We will take a little amount of water. We will pour in that particular pump. Then when we start pumping that particular pump continuously, then we will get water. Correct, na? To get the water, first we will pour little water. Then we will be getting the water. Same way, friends. What is the same way? No change. What is the function of a DNA polymerase enzyme? It has to synthesize the nucleotide. If it want to synthesize the nucleotide, first I have to add the nucleotide. I have to add certain nucleotides. So once I add the nucleotides, to that nucleotides only the DNA polymerase enzyme will come and synthesize or add continuously. So friends, what I am adding? I am adding the primer. who is going to synthesize this particular primer? This primer can be synthesized by a very important enzyme which we call, my friends, primase enzyme. And all the students has to remember one important point, friends. Primase enzyme synthesizing the primer. The primers are what kind of nucleotides? They are not deoxyribonucleotides. They are ribonucleotides. They are RNA molecule. Don't forget this unique statement. That's why, my friends, we can call the primase enzyme is a type of RNA polymerase enzyme. Most of the students will make this particular statement is wrong. When they say that the primase can act like a RNA polymerase, people will say that how primase can act like a RNA polymerase. It's true. Because what is this primer? This primer is basically a polynucleotides of a polyribonucleotides. Remember that particular statement. Now you can ask me, sir, what is the length of this primer? Can you tell me? All of you listen, friends. The length of the primer will be 60 nucleotides. Where in vitro means within the laboratory. Friends, now you people quickly recall our PCR technique. In the PCR technique, what is the first step? First step is the denaturation. What is the second step? Annealing. Correct na? So during the annealing, we will add the primer, right? That primer length is actually 60 nucleotide. But now, sir, if you ask me, sir, what is the length of primer in our body? I can tell you, friends, the length of primer in our body plus or minus 12 nucleotides. That's all. Plus or minus 12 nucleotide is the length of the primer in case of a prokaryotic organism. Sorry, in the body, and in case of in vitro or within the laboratory, friends, it is 60 nucleotides. Don't forget that. Now, one more very interesting point. Sir, how many primers we have to add in the leading strand? Always remember, every student, in the leading strand, friends, we are going to add only one primer, just one primer. Once we add the primer, obviously polymerase enzyme will come and synthesize that continuously. But my friends, when you look into this particular leading strand, what is the statement I want you to remember? In leading strands, look into this. One primer, Okazaki fragment, primer, Okazaki fragment, primer, Okazaki fragment. So all the student has to remember, in case of a lagging strand, many primers are present, but in case of a leading strand, friends, there is only one primer will be present. Never ever forget that particular statement. Now, my friends, all of you listen. Whatever the arrow I am showing to you, this is the direction of the replication fork. Correct na? So one strand will move exactly towards this. You only look into the arrow very clearly. The strand which is moving towards the replication fork, we can call the leading strand. And this kind of a synthesis only, my friends, we will be calling it as the continuous synthesis. But friends, if you look into the other strand, it is moving away from the replication fork. Fork is moving like this. Strand is synthesizing like this. That's why we can call the lagging strand. We can also call it as what, friends? Discontinuous synthesis. What synthesis we call, my friends? Discontinuous synthesis. Don't forget that. So friends, this is what the rules of the DNA replication. So we successfully completed the rules. Now what we have to read? Every single enzyme very depth. Okay. Now everyone just say quickly yes or no. Is that clear, everyone? Now you can ask all your doubts. Till the rules of replication. Fast, friends. Clear, everyone? I hope it's clear for everyone. If you find any kind of a doubts, friends, you can ask. Can we go ahead? Clear, everyone? Okay, perfect. Okay, so friends, now we are going to read what, friends? Enzymes involved in the process of replication. So you all commonly know, right? Most of the students, what they will do, Most of the students, they just read all the enzymes and they just read their functions and just they leave that. That is not how you have to prepare for the CSIR exam. When you are reading for one particular enzyme, you have to read that very briefly, starting from structure. Every enzyme structural arrangement you have to understand. Most importantly, function you have to understand. The most unique statement, whether this particular one enzyme can be connected to the another enzyme or not. That also we have to read very, very elaboratively in this particular enzymology. Both Okazaki and primer. Correct. Correct, Tarun. In case of lagging strand, more number of, see, it's like this: one primer, one Okazaki, one primer, one Okazaki, one primer, one Okazaki. It's like that. Okay, I'll show you in the process. You will get a crystal clear idea. Now let's talk. First, in just 1 minute, I will tell you what are the functions of enzymes in 1 minute. So this is the double-stranded DNA, we know very well. So if the double-stranded DNA wants to open the DNA, sorry, if this particular double-stranded DNA wants to get opened, so what is the first enzyme? Helicase. All of you keep it in your mind. The helicase, in case of a prokaryotic organism, friends, we will be calling it as the DNAB. Don't forget that. In prokaryotes, you know, friends, in eukaryotes, anyway, I will tell you later. In eukaryotes, we can call helicase as a MCM protein. Don't forget that, friends. So what is the function of helicase? It opens the double-stranded DNA. Very simple. When it opens the double-stranded DNA, all of you listen to me. So all of you understand. I'll explain once again in the upcoming slide. When two strands are opened, there is a chance. What is the chance? Once again, two strands can rejoin with each other. Two strands can reannul with each other. Friends, how can we break the strand? We will just break the hydrogen bonds. Joining the hydrogen bond is not that much difficult, right? So once again, there is a chance that the two strands can rejoin with each other. So to prevent the rejoining, to prevent the reannealing, we will use a very important enzymes. What do you call it as, friends? SSB, single stranded binding proteins, which are involved in the stability, means which never allow the two single strand not to associated with each other. After that, now let me tell you one very interesting point. What is that interesting point? Friends, the helicase enzyme will move very faster. You know how much fast it will move? Helicase enzyme will move 1000 base pairs per second. In 1 second, it will break 1000 base pair. That much fast it is moving. So whenever it is moving that much fast, it creates a great tension on the DNA. It creates a great pressure on the DNA. Because of that pressure, DNA undergoes what, friends? DNA undergoes the process of coiling. So to remove that particular coiling, what kind of unique enzyme we required? Topoisomerase enzyme we required. In prokaryotes, we will widely call it as gyrase. I will explain very depth. You just listen. This is the overview. Now, finally, what is happening? Primase enzyme will come and add the primer. Ultimately, the versatile DNA polymerase enzyme, what it is going to do? It will come and synthesize the new strands. Finally, primers can be removed by the polymerases only. Ligase will come and seal the nick. Now that is the overview. Now let's talk about one by one enzyme very clearly. Look into this very quickly, friends. So helicase is the enzyme which will separate the two strand. Single strand binding proteins, they prevent the reannealing or the rejoining. Topoisomerase prevent the torsion. Torsion means stress, coiling by breaking of the DNA. We will read how. Then followed by the DNA polymerase enzyme. Sorry, before the DNA polymerase enzyme, primase enzyme will add the primer. DNA polymerase just elongate that. Finally, my friends, ligase will come and seal the nick. That's the simple step. Now let's talk about one by one enzyme very elaborately. Whichever I say to you, remember that without fail. Don't skip even a single point which we discuss. I just said to you, DNA helicase in prokaryotes we call DNAB. Now my question is very simple to all of you. This is the DNA, right? This is the DNA. Large molecule DNA is All of you listen. This is the DNA B. This is the helicase protein or the DNA B, and this is our DNA. You only answer my question. Helicase is the one which will open the double-stranded DNA. That is true point. From where the helicase has to open the double-stranded DNA? From a point called as ORC. Now the question is very clear. How does this particular helicase enzyme comes to know where this particular ORC is present on this particular DNA? That is the unique question. What is the question? How helicase enzyme comes to know, how the helicase enzyme recognize where exactly the ORC present on this long molecule of DNA. It cannot do actually. So it require a protein called as a DNA protein. I can say this, please listen my friends. DNA protein, I can call it as a initiator protein. What protein I will be called as? Initiator, which recognize the ORC. So DNA A recognize the ORC, then the DNA B will come and open the double-stranded DNA. Now one more interesting point. They can ask a question in the exam saying that DNA B or helicase is an ATP dependent enzyme. Absolutely true statement. There is no doubt about it. And friends, how this particular helicase will get the ATP? By another protein. We will be calling it as what, friends? DNA C. What do you call this particular DNA C? DNA C we will be calling it as a ATPase. What do you call it as? ATPase. ATPase is the one, all of you friends, ATPase is the one which will provide the energy for the DNA B helicase. Then with the help of DNA A, it recognizes the ORC, then start opening the double-stranded DNA. On summary, if the DNA B wants to show their mechanism of action, it is compulsory. There should be a requirement of the DNA A and there should be a requirement of the DNA C. Without DNA A, without DNA C, DNA B cannot show their function. And I have already told you friends just now how much fast this will move. This DNA helicase enzyme will move 1000 base pair. How many base pairs, friends? 1000 base pairs in second. 1000 base pair per second. So that much fast it's keep on moving. Now all of you please kindly look into the picture friends. This is the helicase. You can see, right? You all has to remember one common hint. Whichever the direction helicase is moving, that direction only you can call the replication fork. That direction only you can call the replication fork. So whichever the direction replication fork is moving, if the new strand is synthesizing in that direction only, you can call leading. This is the direction fork is moving. If any strand is synthesizing oppositely, you never called leading, you will be calling this as the lagging strand. So this is the simplified function of the helicase enzyme. Now all of you pay attention. Now I said to you very clearly that DNA B is what? Opening 1000 base pair per second. So it create a great, it will move with a very high pressure. Two strands will get separated. Now all of you listen. What are the major functions of SSB? Three functions are there. I want you people to remember three functions. First, just listen to me. Two strands got separated like this. There is a chance. What is the chance? Two strands can rejoin. Correct, na? Second point, the two strands are very free, right? Very free. There is a chance that many vulnerable enzymes will come and degrade that. So that's why SSB will help. Anyway, let me write. This is one strand, this is another strand. There is a chance that two strands can rejoin. So why we require SSB? We require SSB first function. It will prevent reannealing. You can also call, my friends, rejoining. No, don't worry about that. Second function. I told you now friends, this DNA molecules are not protected, right? If they are not protected, there is a chance certain vulnerable enzyme will come and degrade this particular unprotected single-stranded DNAs. That time, what the SSB will do, it will prevent degradation also. If the SSBs will be associated with the single-stranded DNA never ever get degraded. Third unique function. Now this is the single stranded DNA, right? You know, right? The single stranded DNA, there is a chance that it can form a hairpin loop like structures. They can form a coiling. So what is the third function? It will prevent coiling. It prevent what, friends? Coiling. So the three major functions of the SSB, the ultimate function is what? Prevent the rejoining or reannealing, prevents the degradation. Third one, prevents the coiling also. Now you can ask me, sir, total how many number of SSB proteins will associate? I can tell you very clearly, total 60 number of SSB proteins will be associated. Here I would like to say one more point about the SSB. All the SSB proteins work in a cooperative fashion. What do you mean by that? What do you mean by the word? They will be work in a cooperative fashion, which means, listen to me. This is one SSB, all of you, let me draw here. This is one SSB. One SSB immediately provide the binding site for another SSB. Second SSB will provide the binding site for the third SSB. Third will provide the binding site for 4. 4 will provide the binding site for 5. Like this, friends, nearly 60 SSB molecule will associated with the single stranded DNAs and will do these three major functions. Very simple it is. Okay, friends, is that clear everyone till here? I hope every student is clear till this particular slide. Say yes or no. Okay, and once again I am telling you, I am giving you 1000% assurance. You will get a crystal clear concept once I finish the lessons. Okay, in the middle we can't understand anything because this molecular biology chapter, immunology chapter, cell signaling chapter, they are all interconnected. Once we connect all, then we can come to a conclusion. Then we will become expert. Okay. Okay, I hope it's clear for everyone, friends. Okay, that's all for now. I will meet in the tomorrow sharp 8:30 p.m. What we are going to discuss tomorrow? We are going to discuss the photosynthesis class 2. Okay, and one more important point, maximum by Friday or by Saturday morning. Maximum by Friday or Saturday morning, we will be having 6 to 7 a.m. class, 8:30 to 9:30 p.m. class. Okay, I request please try to attend 50 students continuously. Okay, I am requesting please the only request from my side is please forward to more number of students, by that everyone can get benefited. That's the only my intention. So I am requesting all of you please forward, and one more important one, please kindly give your comments, by that I can improve if you are expecting something else. Okay, friends, 60 SSB on leading and lagging separately. Correct, Priya? In the leading also 60, lagging also 60. Okay, friends, that's all for now. I'll meet you in the tomorrow sharp 8:30 p.m. So tomorrow afternoon you can find this recorded video on the PPT presentation in the website. You can download it. Bye-bye. Good night.